And we're looking at fungal contamination of pharmaceutical products. Okay, so this presentation is going to look at the fungal contamination of pharmaceutical products, and specifically we're going to have a look at the recalls associated with sterility assurance issues, and also some recalls associated with non-sterile products. We'll also have a look at fungal contamination sources in clean rooms, have a look at the detection of fungi and environmental monitoring. We'll also have a look at some approaches that can be taken for fungal identification, and also have a look at why the characterization of fungi matters as part of a identification system. And there's also going to be reference throughout the presentation to some literature. If that's literature that you're interested in reading, then Pharmig can provide some copies. If you could email Maxine or email info@pharmig.org.uk, then we'll be happy to send you copies of some or all of the papers that are referenced. Okay, so let's begin with looking at pharmaceutical product recalls. So the data that's presented on the screen is data that's been extracted from FDA warning letters. And the reason that it has a North American bias is because under the Freedom of Information Act, this information is far more readily available than it is from within the European Union. But I think it captures some general trends. So the data that's captured is looking at a 20-year period, and that's divided up into two sections. First section is looking at 1990 to 1999, and the next section is looking at from the year 2000 up to 2010. And what this data is telling us is that the number of recalls has increased in general due to microbial contamination. And that's a trend that's carried on into recent years, as any kind of scanning of FDA warning letters indicates. The data also tells us that the number of recalls relating to fungal contamination has also risen. Now, of the different microbiological recalls, while the number one reason for recall remains contamination with gram-negative rods, often due to some form of water contamination, fungi now occupy second place, and they account for in the region of 21% for all microbiological related contamination recalls. So fungi account for a major reason for why pharmaceutical products are recalled from the marketplace once they've been released. Now, of the different recalls, some from literature have been considered to be a major risk in relation to potentially causing patient harm. And amongst these are human tissue material, antiseptic mouthwashes, tattoo ink, baby lotion, contact lens solutions, skin moisturizing lotions, and gout treatment. And there's also some other things also listed on the slide as well. Some of the others were not so much a risk of direct patient harm, but still could lead to spoilage of the pharmaceutical preparation. Now, of these recalls, as I said, a number can be considered a risk to patient harm, and the more serious ones are listed on the slide. And drawing from the table, some of these products were leading to infections that could potentially have a high mortality rate, particularly for people who had suppressed immune systems or immunocompromised in some way. And some of the fungal species that were recovered are listed on the slide. And they're important to note because when we come to have a look at common cleanroom fungal contaminants later, a pattern begins to emerge. Now, in addition to those recalls, there's been one major incident in recent years of fungal contamination. And this is the incident surrounding the New England compounding pharmacy. Now, a compounding pharmacy, as I'm sure many of you are aware, is a pharmacy that takes someone else's medicines and repackages them. In the case of the New England compounding pharmacy, this was taking a product that was originally filled in vials and putting it into syringes. Now, a series of incidents took place at the New England compounding pharmacy, and this led to a contaminated sterile steroid product being distributed across 23 US states. And this product was intended as a cure for arthritis. Now, the product was contaminated with fungi, and this contamination caused fungal meningitis, leading to a swelling of the protective membranes that covered the brain and the spinal cord. Now, according to figures that were collated by the US Centers for Disease Control and Prevention, there were 751 people who became infected in 20 states, and 64 people died, and the remaining patient population suffered ongoing injuries. And this stands as the most serious sterile products contamination event ever recorded. The primary contaminant was a fungus called Exocerilium rostratum. They also found Aspergillus fumigatus, but it was the Exocerilium fungus that caused the deaths and the crippling injuries. Now, Exocerilium is a common mold found in soil and on many plants. It thrives in warm and humid climates, and it appears as a black mold when it's visible. So it's what's called a melanized fungus. The risk to the immunocompromised patient is infection of the sinuses, the eyes, the lungs, the lining of the heart, and the bone. And for someone so infected, symptoms will appear between 1 to 4 weeks following a bloodstream infection. Now, this incident occurred in 2012, and on the 6th of October, the pharmacy announced a recall of all its products, a total of 9 different medicines. And this led to a special hearing in the Senate and subsequent legal action. And the issue remains topical because only 2 or 3 weeks ago, the chief executive of the company was imprisoned for the infections that were caused, and the chief pharmacist is still on trial in the US as we speak. Through the infection, over 13,000 units of the contaminated drug were distributed. The root causes were contamination in the aseptic processing area. So when state pharmacists and the FDA went in, they found mold on the HVAC grills leading into the clean room. There was also practice of inadequate cleaning and disinfection to the extent that staff were sometimes walking through puddles of water in corridors to access clean rooms. The sterilisation cycles for the autoclave were unvalidated, and some of the equipment was shown to be producing high levels of particulates. But it appeared that the key root cause was problems with the HVAC system. The pharmacy was next door to a landfill site, and the air system was bringing in fungal spores, and the HVAC system was not capable, and the HEPA filters were not robust enough to eliminate all of that fungi. So that's an important note, and if ever the question emerges, is there a risk of fungal contamination? This is a very pertinent case study to keep revisiting. Okay, so with these types of incidents I've mentioned, we know that fungal contamination is serious. Now, any contamination in a pharmaceutical product has a risk attached to it. With sterile products, all instances are always going to be high risk. With non-sterile products, then any contaminant needs to be risk assessed. And in terms of risk assessing a non-sterile product, then there is a standard schema that's involved, and this was put together by the late Scott Sutton, and it's still one of the best risk assessments for looking at non-sterile products that I've personally come across. And that involves looking at the absolute numbers of microorganisms that are seen, reviewing the characteristics of the microorganism, carrying out literature reviews to ascertain whether the microorganism is a known pathogen, evaluating whether that microorganism has the potential to cause spoilage, then assessing the formulation and characteristics of the product, and then risk reviewing the patient population. And that's looking at how susceptible the intended patient is to the risk. So there are here significant differences, say, between an adult and a child, or someone with a weak or suppressed immune system. So that approach by Sutton is always worth referring to, and if you're not familiar with it, it's available online. Okay, so at this stage, let's briefly pause to consider fungi. So fungi are classified as a kingdom, and that's a separate kingdom to plants, animals, and to bacteria. All fungi have cell walls that contain the material chitin. So this is the thing that differentiates fungi to, say, plants. Plants would have cell walls composed of cellulose. And there are approximately 1.5 million species of fungi. And these are divided into those that produce opaque, creamy, or pasty colonies, and these tend to be called yeasts. There are those that produce cottony, woolly, fluffy, or powdery aerial growths, and these are called moulds. But sometimes this distinction between yeasts and moulds is not always clear cut. So if you take what's generally regarded as a yeast, Candida albicans, it has the ability to form hyphae. There is a third category, mushrooms and toadstools, but if that's a concern in pharmaceutical processing, then something seriously has gone wrong. So fungi are a concern because they are very hardy. They can grow in a range of habitats, including what's regarded as extreme environments, so either very wet, very dry, or having low, sometimes higher temperatures. And they can also readily spread spores over a fairly wide area. Now, it's estimated that about 50% of the known fungi have the potential to cause infections in people. And an infection from a fungus is known as mycosis. In relation to non-sterile pharmaceuticals, fungal contamination of some products is perhaps more serious than others. So there are risks from air, such as the inhalation of spores that perhaps might come from an inhaler. And there are risks from skin, particularly the rubbing in of creams and ointments if the skin is abrased, cracks or bleeding. The risks are increased by the ability of fungi to grow, and many fungi are quite proficient at living off keratin, and this is the protein that makes up the skin, hair and nails of the human body. But people are more likely to get a fungal infection if they have a weakened immune system or if they're going through a course of antibiotics. And in relation to treating fungal infections, these are notoriously difficult to treat in some circumstances. Now, linking back to pharmaceutical recalls. So with pharmaceuticals, most recalls are related to filamentous fungi rather than yeast. And the filamentous fungus is what's commonly called a mold. As these fungi grow, they form branches or hyphae, which eventually come to form a vast network of hyphae, which is called a mycelium. And on an agar plate, that will begin to appear as a visible colony. Such fungi also form spores, and the spores of these fungi have a large range of sizes, although the range of the sizes and perhaps the distance they might be able to travel and how they're carried in airstreams and so on is dependent upon the species, it's dependent upon the prevailing environmental conditions and perhaps the age of the organism. And even within the same species, the spores can vary. So fungi can have a narrow range of spore sizes, such as with Aspergillus niger, or they can have a wide range of spore sizes, as might be found with some species of penicillium. Okay, so what in particular should we be concerned about? Well, from this review of pharmaceutical recalls, a relatively few number of species make up the vast majority of recalls. So of the 1.5 million potential species of fungi, there's a handful that pose a risk to pharmaceutical products. And these are, and I'm going to go through these in the order that they've occurred in recall lists. So starting with Cladosporium, Cladosporium has been responsible for the vast majority, that's about 30% of the recalls. And Cladosporium is a very common indoor mold. It's commonly found on living and dead plant material, and it particularly likes damp conditions. So as it's found throughout many homes, it's the same type of environmental fungus that would pose a risk in a less well-controlled pharmaceutical facility. Next on the list, accounting for 25% of the recalls, is penicillium. And this is a common soil fungus. And this prefers cool and moderate climates. It's also a common spoiler of food. Next comes Aspergillus, and 20%. And this is very common to most environments. And Aspergilli are common contaminants of starchy food, such as bread and potatoes. Some species can also cause quite serious diseases in humans and animals, and that's because they produce toxins called aflatoxins. And a species called Aspergillus flavus is a notorious example of that. It's responsible for a disease that's common with agricultural workers, the condition of farmer's lung. Only for 10% of the species is Alternaria, And this fungus leads to common allergens in humans. It's often found growing indoors, and it can trigger reactions like hay fever or the hypersensitivity reactions that could sometimes trigger asthma. Okay, and then coming in also at 10%, we have trichophyton. Now, trichophyton species are the most common cause of athlete's foot, as well as ringworm and jock itch and infections of the nail, beard, skin and scalp. Now, I do find this one interesting because trichophyton is quite notoriously difficult to grow on culture media, yet it stands at 10% of the recalls, so quite how it was assessed is an interesting issue. Okay, and then coming in at 5% is Candida, and that's the only yeast listed on the list of recalls. So it goes back to what I was saying earlier about filamentous fungi being the majority. Filamentous fungi accounted for 95% of recalls over a 20-year period. With Candida, this is universal on most normal adult skin, and it is harmless to individuals on skin, but can cause problems if it ends up in inappropriate places. Now, of those fungi that are theoretically posing the greatest risk in terms of their of being pathogenic, there's Candida I've mentioned, and Candida is a significant opportunistic contaminant in immunocompromised people. So, for example, a transplant patient would be at great risk from a Candida infection, someone like Candida albicans. And then Aspergillus. And it's those Aspergillus species that produce the mycotoxins or aflatoxins that can also trigger quite serious allergic reactions. So Aspergillus flavus, as I previously mentioned, and then Aspergillus fumigatus as well is another serious type of fungus. Okay, so now I'd like to consider where fungi can occur in the pharmaceutical environment. Now, like any form of microbial contamination, fungi can be present in clean rooms, and they can present a product risk. So fungi can be associated with the air, be associated with water, be carried by people, they can be present on equipment, and they can come in and out of areas on materials. But perhaps the biggest risk is from air, because fungal spores, which can range in size from 1 to 50 microns, can be easily dispersed into the clean room environment. And then depending upon the air distribution, and given the fact that many clean rooms have turbulent airflow, can be distributed to many parts of the clean room. So we'll have a look at aspects of manufacturing in a clean room in more detail now. So let's first consider clean room design and operations. So a number of aspects of clean room design can release fungi, assuming fungi are present. Or they allow fungi to be transferred in, or they provide opportunities, niches for fungi to grow. So some examples are door kickback plates, particularly if spores are between the plate and the door, bags, incubators, boxes, particularly depending on the material, and another reason in addition to bacterial spores why cardboard should never end up in a clean room. Trolley wheels are a notorious source, partly because invariably people forget to disinfect trolley wheels when moving trolleys between different areas. Loose ceiling tiles, damaged flooring. Also vibrations from construction. So there's been some cases where work's been taking place in one part of the building, not necessarily even in the clean room, but the vibrations through the building structure cause fungi to be dislodged into a clean room. Faulty light fixtures, problems with HVAC systems, say a hole in a HEPA filter or leakage around the HEPA filter housing, and with items going in and out of clean rooms. Okay, so looking at buildings in a little bit more detail. So with walls and ceilings, poorly ventilated areas where there's insufficient air changes will help fungal spores remain in the area. And that's something to take note of. At the moment there's quite a drive in the pharmaceutical industry to reduce the number of air change rates in clean rooms in order to save energy. But you need to be sure that you're not going down too far to lead to poor air distribution patterns. Then there's risks from ridges or cracks in finishes, areas where spores can be deposited, fungi can then start growing. Plaster and paint can be a nutritional source for fungi. So the types of paints used in clean rooms need to be clean room suitable. And incidentally, some research suggests that fungi can grow more readily in matte finishes than they can in high gloss finishes. An alternative is to add a fungus mat to a paint, or a paint that comes formulated with a fungostatic material. So there are paints that can be purchased that contain, say, 1% pentachlorophenol. Now, the main types of fungi that can be recovered from clean rooms are Cladosporium, Aspergillus, and Penicillium. So those three fungi are also the top three associated with pharmaceutical product recalls. So there's, perhaps unsurprisingly, a connection between the fungi most commonly recovered from clean rooms and the fungi most commonly associated with pharmaceutical product recalls. Okay, so now looking at other sources. So raw materials can sometimes carry fungal spores, and fungal flora are more likely to be associated with incoming raw materials of plant origin. So things like gums, agars and starches. And here Cladosporium, Alternaria and Fusarium have been most closely associated with raw materials of vegetative origin. Then there's packing materials. So risk relates to the composition of the material and how it's stored. Risk factors include if there was to be dust on wrapping or on the material itself. Cardboard, which I've said is a big issue, and the way things are transported in and out of clean rooms. And the primary species here are Penicillium and Aspergillus, as well, incidentally, as spores of bacillus organisms. With equipment, filters or straining bags made from natural material, such as paper, canvas, and so on. Another problem is that if there's using a cellulosic material, then fungi can utilise the cellulose and impair the fibres. There's been cases of cellulosic bags or wrapping literally rotting away because of fungal contamination. And the primary species of concern here is Cladosporium. So, for everything discussed so far, the biggest concern is with clean room air. And the problem of air was shown in a big study of HVAC systems. And this was published in the Journal of Occupational Environmental Hygiene back in 2007. And this study, the researchers undertook 566 tape lifts, which is not as sophisticated as it might sound, but actually went round with strips of sellotape. But it was effective, and 570 swabs. And from that, they found a high level of recovery of cladosporium. And it was found in places like blower wheel, fan blades, ductwork, and cooling coil fins. So that was an interesting study highlighting the potential cleanroom risks. So the previous slides are very much about where fungi might be found. Now, of course, all this is potential, depending on whether fungi has or hasn't got into the facility. But also, should fungi get in, the extent that it might cause a problem depends on influencing factors. And these will increase or decrease the likelihood of fungi. So with the list that's on the slide, one factor or a combination of factors will lead to a higher chance of fungal growth. So these factors need to be weighed up are wet or damp areas, where there's changes to environmental conditions, such as an alteration in ambient temperature or change to air humidity, personnel behaviour and hygiene, where there's ineffective cleaning and disinfection, the time of year. So there is some studies that suggest that fungal levels spike in autumn. The geographic environment, so where in the world the facility is, and the level of ventilation and clean room design. Another major source of fungi comes from people. Another comes from fungi that are naturally resident to the body, or where fungals carried on people in a transient state. So we know that the human body contains, inside and outside, lots of microorganisms. And we also know that we shed a lot of skin cells each day, a multi-million number, perhaps even approaching a billion. We know from studies by key researchers like Bill White that about 10% of the shed skin cells contain microorganisms. So within the clean room, they're acting as microbial carrying particles. And we thought, at least until fairly recently, we had a good idea about the human body and microorganisms. And that's shifting a bit now following the information that's coming from the Human Microbiome Project. And the Human Microbiome Project is a major international study looking at the bacteria and fungi carried on the human body. And the Human Microbiome Project has taught us that we are host to a considerable diversity of species. There's also variation between different locations on the body. So skin has a relatively low diversity, but it does have a high population. It's also that the variation differs between individuals, and that our own microbiomes change as we age. So what does that mean for fungi? Well, one major study carried out by a researcher called Finlay showed that with skin locations for fungi, the areas where there's a high number of fungi is the torso and between the toe webs and on the heel. And there's lower numbers of fungi on the forehead, arms and scalp. So that information perhaps is useful for considering personnel gowning and gowning practices if fungal contamination is a concern. The major types of fungi found were Malassarea, which is the dandruff-causing fungus, which hasn't been associated in any pharmaceutical recalls, but also Penicillium and Aspergillus, which have. So, lots of fungi on the skin in different places. But we should be able to control this through good clean room design, good control of air, and through good gowning. And where anything more important needs to take place, through protective airflows, which would apply to steriles and non-steriles, and in sterile pharmaceuticals through the use of barrier technology. We can also assess the risk through a well-constructed environmental monitoring program. Now, along with most aspects of environmental monitoring, regulators express an interest but give very little guidance. So, likewise, regulators express an interest in fungi and with environmental monitoring, but there's no document produced by any regulatory agency that says anything about the types of culture media required, with one exception, which is in the Japanese pharmacopoeia. However, the FDA guidance for aseptic filling, which was last revised in 2004, does state that agar must have undergone growth promotion testing for bacteria and for fungi. So, with most things environmental monitoring related, there's a variety of practice. So some sites use a general purpose culture media incubated at 2 temperatures. This would be probably a higher temperature, so 30 to 35 degrees, to encourage the recovery of skin commensurable bacteria. And a lower one at 20 to 25, perhaps designed to recover fungi and some other bacteria. Other sites choose to use 2 different media. A big limitation in relation to fungi relates to the culture media, types used, and the incubations. Some of the selected media that is sometimes adopted is malt extract agar, which is recommended by the American Conference of Governmental Industrial Hygienists. Sabrose dextrose agar, which is listed regularly in the European pharmacopoeia, not for environmental monitoring, but for various bioburden tests. Potato dextrose agar, and the pink-coloured rose Bengal agar. Now, when picking a selective agar, many people do adopt for Sabrose dextrose agar, and that's because it does appear in the European pharmacopoeia, United States pharmacopoeia, and Japanese pharmacopoeia. But if a selective or general purpose media is used, both do need a degree of qualification. And this needs to look at incubation time and temperature. And there's a big issue if a single general purpose agar is used, because if the temperature is wrong, so let's say the media goes in at too a high temperature, then there's a risk of the destruction of the lytic cellular enzymes that are found in fungi, which could then lead to an underestimation of the amount of fungi. If the temperature is too low, then there's risks of overgrowth of mycelia and the potential obscuring of bacterial colonies. So let's have a look at fungal agars. So a little while ago, I undertook a study looking at different fungal agars. So this looked at clean rooms of a facility located in the southeast of England, and it looked at grade C and D clean rooms. And a number of different media were looked at. So the media we looked at were malt extract agar, a malt extract agar containing antibiotics, potato dextrose agar, rose Bengal agar, and Sabrose dextrose agar. And the aim of this study was to assess if there was any significant difference in the numbers of fungal isolates recovered from these 5 agars, and also to assess any variation in recovery of different species or genre through these selective agars. So the outcome of this study was that recovery of fungi was relatively low, and the mean counts ranged from less than 1 to about 8 colonies per sample type. Interestingly, air samplers tended to record more fungi than surface samples. So that goes back to the earlier theoretical discussion that airborne fungi in clean rooms present the greatest challenge. It was also found that there were far more filamentous fungi recovered from the clean room environment than there were yeast, which also tallies with the list of fungi from pharmaceutical product recalls where yeasts do not feature very regularly. So the results from these different agars were studied for significance testing, and it was found that if yeasts are a concern, then malt extract agar gives the highest recovery of yeast, followed by sucrose dextrose agar. Potato dextrose agar recovered the lowest variety. With filamentous fungi, the optimal media for recovering filamentous fungi was rose bengal agar. Then came sucrose dextrose agar, but the poorest performing one was malt extract agar. So what does this tell us? It could tell us that if we're not really concerned about yeast, then rose bengal agar might be the best one to use. However, that's an agar that tends to come with a short expiry date, needs to be kept in the cold room and so on. Secondly, if we're concerned about filamentous fungi and yeast, as perhaps we should be, then the optimal agar, the one that stands the best chance, is in fact sucrose dextrose agar. So the information from the study might prove useful in building that assessment. Now, this study, if you want to have a copy of it and look at that in more detail, this is one of the papers that we certainly can provide. So if you want to contact Pharmig, then you can read about this in more detail. Okay, so now moving on to identification. So if fungi are reasonably prevalent in clean rooms, and certainly from the recall list, potential contaminants of a range of pharmaceutical products, and if the only way with non-sterile products is to know the fungus in order to characterise the risk, then identification of fungi is a matter of importance. The problem again arises is that there's no standard guideline on types of agars, identification methods. Many commercial phenotypic systems cannot readily identify fungi except for a narrow range of yeasts. Genotypic systems are often expensive and beyond the scope of many laboratories. There aren't that many identification books for fungi that focus on industrial fungi and pharmaceutical fungi. Most tend to veer towards the clinical. And few microbiology laboratory technicians are actually all that skilled in fungal identification. So these are some barriers that exist in relation to fungal identification. So a little while ago, I worked with some microbiologists based in India to see what could be done. So we set out and undertook a review of the primary fungal contaminants in the pharmaceutical industry. And from our literature study, we set out to provide guidance on fungal identification. Our objectives were to use phenotypic methods, to use methods that were not too convoluted in terms of time or money, to provide clear instructions, and only to use agars that were actually commonly available. We also wanted our approach to be suitable for use worldwide. So this involved looking at a number of key steps for the identification of fungi. So first, it was quite clear that the type of agar can influence the fungal identification, and that different fungi grow better on different agars. So one of the things set out to do was to try and match agars with different primary fungal contaminants. Second point was that everything required a standard incubation time, because if there's variations in incubation times, then it would lead to different identification outcomes. So everything that we did was based on 5 to 7 days incubation at 20 to 25 degrees. The third step was to draw up a list of key criteria that should be recorded and observed, so that a laboratory technician can compare their macroscopic observations against observed criteria. So this included things like colour, texture, the presence of a diffusible pigment, whether there's any fluid release, growth zones, the growth of hyphae, whether that was aerial or submerged, rate of growth, the colony topography, and other factors relating to shapes and structures. The next phase looked at microscopic staining. And there are different stains that can be adopted for fungi, but from our experience, the optimal one, the clearest one, was to use lactophenol cotton blue as the standard, and provided a standard formula method. And this is quite good because the phenol will actually kill any live organisms, but also it prevents cell lysis. The lactic acid in the stain is very good at preserving the fungal structure, and the blue is very effective at staining the chitins found in the fungal cell wall. We also looked at different ways of getting a portion of the fungus, that part needed for staining, from the plate to be stained. And we looked at some different methods. So there's direct examination. That's basically putting the plate underneath a microscope, and there's a big risk here of breaking delicate structures because of the closeness of the objective to the plate. We looked at adhesive tape. So clear tape is effective, but it's fiddly, and particularly with gloves, and if gloves are not being worn, then there's risk of infection to the laboratory worker. Or there's a touch tape technique, which is the one illustrated, and that requires more skill, but is actually far more effective. And then the next part was to provide an illustrated guide of all things to observe under the microscope to assist with the identification. And these pictures here are taken from the research paper, and this paper is something that we can, that Pharmig is very happy to pass on to you to have a look at. So you don't need to worry about what's on the slide. If you're interested, then we can give you some literature to have a look at. And then seventh, we provided descriptions and photographs of 12 key fungal genera, and these are the ones associated with the pharmaceutical product recalls and also derived from papers relating to environmental monitoring. And this was accompanied by information about growth media and incubation conditions to allow a matching process to take place. So some examples, again taken from the paper. So the first, we have Aspergillus niger, one of the most common species of Aspergillus. And this, although it's characterized by melanized colonies, it can be tricky to identify at first because the black pigmentation takes some time to form. So you get the idea for each fungus presented. There's an image of growth on agar, accompanied by a microscopic image, accompanied by some more detailed notes. Next example is Cladosporium. And you can see, I'll just flick back. So have a look at Aspergillus and then flick back to Cladosporium, just how different look macroscopically and microscopically. And Cladosporium is very important, being the most common indoor mould, and its ability to trigger allergies. And this produces a characteristic olive green to brown looking colony on a plate. And just one more example, we have penicillin, common in soil, and this again has a very different appearance, both microscopically and macroscopically. So again, just pop back up to Planosporium and to Aspergillus, and you can see how different these three very common types of fungi look. So of course, if money is no object, then there is genotypic identification. Now, genotypic identification now is different for fungi than it is with bacteria. So for bacteria, there's often a big focus on the 16S RNA region of the bacterial genome, and this is because it's a highly conserved region, not prone to phenotypic changes. With fungi, there are two major sequencing targets for fungal identification, and these are the D1, D2 region of the large ribosome subunit, what's called the LSU, and also the internal transcribed spatial regions, which are often abbreviated to the ITS1 and ITS2. And the genotypic methods for fungi are based on PCR amplification and sequencing. Now, this requires very specialised technology, and I'd imagine in most cases something that needs to be outsourced. Another problem with this is that of the 1.5 million estimated different types of fungi, only about 165,000 sequences of fungi are held, and these are held on the International Nucleotide Sequence Database. Of these, only a small proportion have names, and of those, only a small proportion are actually in the databases of commercial genotypic fungal identification systems. So this is why there's still a big reliance upon the visual approach to looking for fungi. Okay, so you may be asking the question, why go to this much effort? Does fungal identification matter? Well, it does to an extent, because even just on the basis that if 90% of clean room contamination is gram-positive cocci, staph and micrococci, then if fungi are found, then it's something unusual, some odd event as a concern, and therefore it's something worthy of examination. And knowing the identification allows the microbiologist to try and determine the contamination source. Where else might this organism have been found? It allows the microbiologist to track contamination through the facility. It allows for the formulation of kappa. It helps with trend assessments. It's quite key to determine the effectiveness of cleaning and disinfection. And also it helps with the verification of culture media. So it's probably a good idea to take some of the most common fungi and to use that to challenge environmental monitoring media as growth promotion ahead of release. Another important area is with challenging disinfectants. Because knowing the main fungi found in the pharmaceutical facility is important in relation to disinfectants. And you need to know that the disinfectants you use can kill fungi. Now, just as an aside plug, Pharmag have recently released the Guide to Disinfectants as an updated edition, and the use of fungi very much forms part of the disinfectant efficacy review of that. Pharmag are also going to be shortly launching a training video about cleaning and disinfection, and that again helps to contextualise the importance of facility control and controlling the environment. So that's my plug over, and then back to the importance of disinfectant testing. So a fungal cell may have an innate ability to present a permeability barrier to one or more biocides. And also it's possible that fungal enzymes may inactivate that disinfectant. So truly understanding this can only be answered through disinfectant efficacy studies. And this can be shown through suspension and perhaps more importantly, surface testing. There's also a way to assess this by determining the minimum inhibitory concentration of the disinfectant. That's more of a applied research tool, but it can also help in working out what the lowest concentration of a disinfectant is that can eliminate or exert fungostatic or fungicidal properties. So just an aside, there was another recent study that was done that looked at 3 disinfectants in relation to fungi. And this looked at a biguanide, which was chlorhexidine, which are types of polymers supplied in salt form. So these disinfectants have a fairly wide spectrum of activity. It also looked at 2 quaternary ammonium compounds. So it was a chloride-based chemical and cetramide. And these were cationic salts of organically substituted ammonium compounds. And this looked at a range of clean room fungal isolates using a microdilution method. And the fungi challenged for this were Aspergillus, Penicillium, Cladosporium, Cavalia, and Alternaria. And in terms of the results obtained, it showed that for all the fungal isolates, the minimum inhibitory concentration range was less than 16 micrograms per milliliter, which is higher than that required for non-sporulating bacteria like Staphylococcus aureus or E. coli. So that was interesting, because that showed that fungi are harder to kill than common gram-positive coccus or a representative gram-negative. However, another bacterium like Pseudomonas aeruginosa proved more resistant to fungal spores. So this is just an interesting example of where fungi may not fit naturally well on sometimes some of the standard disinfectant hierarchies that are presented. But what it perhaps informs is that perhaps periodically fungi as well as bacteria, the most common things found from an environmental program, should be periodically challenged to disinfectants as part of an ongoing review. So just drawing the presentation to a close. So on the slide are 4 studies that I was involved in that the presentation has made reference to. Now, Pharma can provide copies of these to any of you. So, as I said, email Maxine or email info@pharma.org.uk. The first one is a general overview of the types of organisms found in clean rooms. That was a 10-year study. The second one looks at the antifungal efficacy of disinfectants against clean room fungal isolates. The 4th one is the one that we spent most time on. So that's the one that provides a identification guide to the 12 most common fungi found from pharmaceutical product recalls. And the final one is a review of different fungal agars. And that was the one that showed that SDA is the optimal fungus for study. Okay, so drawing this presentation to an end, what we've looked at is recalls of pharmaceutical products due to fungal contamination and which are the most common fungi associated with that. We've looked at the aspects of environmental monitoring and clean room control and which are the most common fungi found in clean rooms, and linked that back to the pharmaceutical product recalls. We've also looked at aspects of environmental monitoring, which is the best agars to use, and the difficulties of fungal identification, and wrapped up to say why fungal identification matters.